everyone, and welcome back to the PFF Summit and the 2021 PFF Scholars Symposium. I'm Michael Beers. I'm a physician scientist at the University of Pennsylvania. I'm also a member of the Medical and Scientific Advisory Council for the PFF and chair of the Research Review Committee for the PFF Scholars Program. So this symposium is near and dear to my heart. The PFF Scholars Program, as you heard earlier in the week from Drs. Lasky and Schmidt, has funded over $4.5 million in projects, and its mission is to really enhance the lives of patients with pulmonary fibrosis through creating a better understanding of all aspects of the disease. And so from that perspective, I think that you'll find that the symposium today is quite representative of the work that the PFF has done over the last 10 years. The three speakers you're going to hear today are early career investigators working on various different aspects, everything ranging from translational lung science to understanding some of the trials and tribulations associated with being the caregiver for these patients. And I'd like to introduce each of the three of them prior to their presentations. And of course, we'll be back for questions and answers, and please put these into the chat, and we will do our best to answer all the questions at the end of the three talks. So the first speaker is going to be Jillian Gooby, who trained in Canada, but is at the University of Pittsburgh now, where she's actively pursuing a PhD in human genetics, but she's also a attending physician at the University of Pittsburgh. She has an interest in air pollution, and the title of her talk is going to be today, Air Pollution Impacts on Patients with Fibrotic Interstitial Lung Disease. This will be followed up by Dr. Bridget Graney, who's an instructor in the Division of Pulmonary Sciences and Critical Care Medicine at the University of Colorado. She trained in Nebraska and has graduate medical training in both pediatrics and internal medicine and a fellowship in pulmonary and critical care. Her current research focuses on all of the dynamic interrelationships between caretakers and patients with interstitial lung disease. And her talk will focus again on the challenges associated with being a caregiver for patients with interstitial lung disease and pulmonary fibrosis. And then finally, from Tel Aviv, we have Rami Unterman, who trained in Israel and then came to do research with Naftali Kaminsky at Yale University. He has returned to the Saraski Medical Center in Israel, where he runs a research program looking at the genomics of interstitial lung disease. And he's going to share with us his data on single cell profiling of blood immune cells in fibrotic lung diseases. And so with that as an introduction, we'll kick off the first talk with Jillian Gooby from the University of Pittsburgh. Thank you.
Hi, everyone. My name is Jillian Gooby, and I'm a Canadian-trained internal medicine and respirology physician currently undertaking research training as a PhD candidate in the Department of Human Genetics in the Graduate School of Public Health at the University of Pittsburgh. I'm also co-appointed as a PGY7 Research Fellow within the Clinician Investigator Program at the University of British Columbia, and I have the honor of being a Pulmonary Fibrosis Foundation Scholar for the 2020 to 2022 period. Today, I want to talk to you about a part of my PhD research entitled Air Pollution Impacts on Patients with Fibrotic Interstitial Lung Disease, wherein I will discuss our preliminary data evaluating the impact of air pollution on clinical outcomes in a cohort of patients with ILD from the University of Pittsburgh, and I'll also introduce my future plans to investigate how epigenetic alterations may mediate the adverse outcomes associated with air pollution in this population. In terms of my disclosures, I'm the grateful recipient of funding from the Pulmonary Fibrosis Foundation Scholars, the UBC Clinician Investigator Program, and the UPIT Pulmonary Allergy and Critical Care Medicine Discovery Award. So the first message I want to convey to you is that air pollution is a massive global health problem. Over 90% of the world's population live in areas where air pollution exposures exceed WHO recommendations. And PM2.5, or particulate matter with a diameter of 2.5 micrometers or less, has been estimated to cause about 8.9 million premature deaths annually. And so the figures depicted here demonstrate that as PM2.5 levels increase, the hazard ratio for all cause and disease-specific mortality also increases. And you can see on the plot on the right here that a large burden of the cause-specific mortality from PM2.5 manifests as respiratory disease, including COPD in purple, lung cancer in yellow, and lower respiratory tract infection in black. But what's not shown here is my disease group of interest, which is interstitial lung diseases, or ILDs, for which there's less of a clear picture of the impacts of air pollution. So as most of you are already familiar with, ILDs are a group of over 200 pathologic entities that are characterized by scarring of the lungs, as well as clinical symptoms of dyspnea, cough, and progressive respiratory failure. These diseases have a universally high morbidity and mortality, with the most common form of ILD, which is idiopathic pulmonary fibrosis, having a median survival of 3 to 5 years from the time of diagnosis. And so my interest in ILDs is related to the intersection between sociodemographic, environmental exposures, and epigenetic factors in these patients. And in order to summarize the literature to date and to highlight the potential mechanisms underlying air pollution's adverse impacts in patients with ILD, we recently published this article in the American Journal of Respiratory and Critical Care Medicine, which reviews the studies linking air pollution to adverse outcomes in patients with ILD and outlines how epigenetic changes may mediate these outcomes. To summarize the knowledge presented in that review article, there is evidence to indicate that airborne pollutants are associated with preclinical ILD, in other words, interstitial lung abnormalities, and possibly the incidence of IPF. Other studies performed in patients with IPF have demonstrated that a variety of different pollutants are associated with adverse clinical outcomes, including acute exacerbations, lung function decline, and mortality. But many of these studies are limited by some common factors. So there's minimal consideration of non-IPF fibrotic ILD patients. These studies generally investigate outcomes in a relatively limited geographic region and isolated populations. Many of the techniques that are used in these studies for estimating air pollution exposure are fraught with exposure misclassification issues. Many of these studies do not consider other environmental or socioeconomic factors such as neighborhood level disadvantage. And the mechanisms underlying the association between air pollution and adverse outcomes in these patients has also been minimally explored. So we aim to target some of these limitations of previous studies with our research here in investigating the impact of air pollution in patients with fibrotic ILD through these central hypotheses. So we hypothesize that higher residential air pollution exposure, specifically PM2.5 and its constituent components, is associated with increased mortality, worse baseline lung function, more rapid lung function decline, and alterations to DNA methylation patterns in peripheral blood samples from patients with fibrotic ILD, which may mediate the adverse clinical outcomes noted above. And this work goes beyond previous studies, as it will evaluate the impact of multiple pollutants on multiple clinical outcomes in a highly diverse cohort of patients with fibrotic ILD. Today, I'm just going to be showing some of the results from the Simmons Center cohort at the University of Pittsburgh, reflecting around 1,400 patients. But I will be performing these same analyses on another 2,000 patients from the PFF registry and another approximately 4,000 patients from the Canadian Registry for Pulmonary Fibrosis in Canada, which will make this the largest evaluation of air pollution impacts in patients with fibrotic ILD to date, and the only to look at fibrotic ILDs as a group rather than specific diagnostic subgroups. We also use a novel and precise methodology for evaluating air pollution exposure that provides substantial improvements over previous exposure estimation techniques. And this will also be the first series of translational studies to link air pollution and clinical outcomes with epigenetic and other molecular markers of disease in these patients. So to perform my air pollution exposure calculations, I'm using a robust data set that is freely accessible through the Atmospheric Composition Analysis Group at Dalhousie University. And they use a satellite hybrid technique to estimate air pollution exposures of particulate matter through a measurement called aerosol optical depth, which is essentially the visual opacity over a region that is not due to cloud cover. And they combine these estimates with chemical transport models and ground-based measurements of pollutants, such as through Environmental Protection Agency ground-based monitors, to come up with a highly accurate and precise estimate of PM2.5 pollution at any region throughout North America. And this technique has a number of strengths. Most notably, it has excellent spatial resolution, where estimates are accurate to approximately 1 kilometer squared, which is much better than most other exposure estimation techniques. And these models are also able to consider topography and meteorology in their calculations. So here I'm showing an example of the satellite-derived PM2.5 data for the year 2005 that we were able to compile using the Dalhousie University data. And you can see that there's excellent resolution across the US and Canada, and that in the Western Pennsylvania region where I'm currently living now, there were quite high levels of PM2.5 during that year, generally in the range of 12 to 16 microgram per meter cubed, which is much above the WHO and EPA recommended exposure levels for average yearly exposure to PM2.5. And if you look over at LA region, this is where the PM2.5 levels were highest in that year on an average of about 20 microgram per meter cubed. And the other interesting thing to note is that there are spots of red in sparsely populated regions in Quebec and Alaska. And these are actually reflective of wildfires that occurred in that year. So this highlights how this type of exposure estimation technique can capture the regional and temporal variations that are often not captured by traditional techniques, especially in rural and sparsely populated areas. So now I'll move on to discuss the results of our preliminary analyses in the Simmons Center cohort of patients with fibrotic ILD at the University of Pittsburgh, where we investigated the impact of satellite-derived PM2.5 exposure on mortality, baseline lung function, and lung function decline. And just to remind everyone, these are preliminary and unpublished results that require further validation with the PFF and the CarePF cohorts prior to publication. And these are analyses that I'll be performing over the next month. So here I'm just showing the baseline characteristics of the Simmons Center cohort that we've evaluated in these analyses. So this is an older and predominantly white male cohort, and we expect to have more sex and race diversity with the inclusion of our other two cohorts later. About 50% of the patients have a diagnosis of IPF with connective tissue disease ILD and unclassifiable ILD as the next most common diagnoses. The median follow-up duration in our cohort was 4.5 years. Here, I'm showing the PM2.5 exposures averaged over several time periods of interest according to our monthly satellite data, which is matched to patient residential addresses. On the left, you can see that the average exposure in the 5 years preceding the diagnosis of fibrotic ILD sits above the WHO standard, but slightly below the National Ambient Air Quality Standard of average yearly PM2.5 exposure, which is a standard that's set by the Environmental Protection Agency. And it's important to note that this EPA NAAQS standard is much higher than most air pollution and pulmonary experts would like it to be. And there's a movement to lower this EPA standard to 8 microgram per meter cubed. So in the 5 years preceding diagnosis, our patients had a median exposure of 11.4 microgram per meter cubed. The middle is showing the average exposure to PM2.5 during the course of disease with fibrotic ILD. And so this is from the time of diagnosis until the time of censoring. And censoring here is defined as death, lung transplant, or cessation of follow-up, either due to the patient being lost to follow-up or due to that being the final period of data collection. And our median exposure during this period of time was 9.7 microgram per meter cube. And on the right, I'm showing the average exposure to PM2.5 in the 5 years prior to censoring. And the median exposure during this period was 9.4 microgram per meter cube. So the 5 years prior to censoring is capturing from that last period of follow-up and the average PM2.5 exposure in the 5 years leading up to that last follow-up or censoring time point. And that's an important period to capture because that may have the greatest implications for disease progression and pathophysiology. And that may or may not include the entire time period of which that patient has had a diagnosis of fibrotic ILD. We wanted to look at these multiple time periods in order to consider the sensitivity of our outcomes to multiple time periods. So I'll show that a little bit more in our data that we're showing later. So here I'm showing the PM2.5 exposures over time in patients with fibrotic ILD at the Simmons Center. And this is their PM2.5 exposure during the course of their disease. So each dot on this scatter plot reflects one patient, and the y-axis is the PM2.5 exposure average over the course of their disease. And what you can see here is that there appears to be a decrease in the average exposures to PM2.5 during the course of the disease over time. So patients that were diagnosed earlier tended to have higher exposures to PM2.5 during their disease as compared to patients that were diagnosed later. And this is likely reflective of improving air quality over time, especially in the Western Pennsylvania region where the Simmons Center resides. And this may be due to decreasing coal and industry-related pollution. Now I'll move on to discussing the mortality analyses, where we use Cox proportional hazards models with death and lung transplant being considered as a composite mortality outcome. So here I'm showing a Kaplan-Meier curve of PM2.5 in the time period of during disease, so from diagnosis until censoring. And we can see that patients in quartile 3 and quartile 4, indicating the highest exposures to PM2.5, had a significantly higher hazard ratio for mortality compared to patients in quartile 1, which is shown in yellow up here. And the interpretation of the continuous PM2.5 hazard ratio here of 1.11 is that for each 1 microgram per meter cubed increase in average PM2.5 exposure during the course of disease, the risk of mortality increased by 11%. And this model is adjusted for sex, age at diagnosis, smoking history, race, neighborhood level disadvantage, and baseline forced vital capacity and diffusion capacity for carbon monoxide. Next, we're looking at the different time period of the 5 years prior to censoring. Again, censoring is the period of death, lung transplant, or cessation of follow-up. And we can see that the separation of the curves on the Kaplan-Meier curve seems even more pronounced than on the previous slide. And we see that there is a significantly higher hazard ratio for mortality in quartile 2, 3, and 4 in comparison to quartile 1, with patients in quartile 4 having approximately 6 times higher mortality risks than patients in quartile 1. And the interpretation of the continuous hazard ratio here of 1.24 is that for each 1 microgram per meter cubed increase in PM2.5 in the 5 years prior to censoring, the average risk of mortality increased by 24%. And we adjusted this model for the same covariates as before. The last time period we looked at was the 5 years prior to diagnosis with fibrotic ILD, and we can see that there is a much less visible difference between the mortality curves. On our Cox models, patients in quartile 2 and quartile 3 had significantly higher hazard ratio for mortality compared to quartile 1, but patients in quartile 4 did not have any significant difference in mortality risk, and there was no significance in the continuous hazard ratio. This was adjusted for the same covariates as before. So this demonstrates that the at-risk period may not be in the 5 years preceding diagnosis. So to summarize our mortality analyses, we found that higher PM2.5 exposure during disease and in the 5 years prior to censoring is associated with significantly increased risk of mortality, but the exposure in the 5 years prior to diagnosis has a less clear impact on survival. Next, I'll move on to discussing the lung function analyses, beginning with the association between pre-diagnosis PM2.5 exposure and the baseline measures of forced vital capacity and diffusion capacity for carbon monoxide using multiple linear regression models. And then I'll show the rate of lung function decline, which we assessed using linear mixed effects models with random intercepts and slopes. So in these multiple linear regression models adjusted for the covariates of age at diagnosis, sex, smoking history, race, and neighborhood disadvantage, we found that living in the highest quartile of PM2.5 exposure in the 5 years preceding a diagnosis of fibrotic ILD was associated with a lower baseline FVC, or forced vital capacity, and that's shown by this significant beta of negative 5.45, which can be interpreted as, compared to quartile 1, patients in quartile 4 of PM2.5 exposure in the 5 years leading up to diagnosis experienced a 5.45 unit lower percent predicted FVC at baseline. There was also a significant association between the continuous PM2.5 in this 5 years pre-diagnosis exposure and baseline FVC with a beta of negative 1.0, indicating that for each 1 microgram per meter cube increase in PM2.5 in the 5 years pre-diagnosis, the baseline FVC decreased by 1% predicted. In the models looking at baseline DLCO, or diffusion capacity for carbon monoxide, adjusted for the same covariates, we did not find any significant association between the quartiled or continuous PM2.5 in the 5 years preceding diagnosis and baseline diffusion capacity. Now I'm moving on to look at the impact of air pollution on rate of decline in lung function. So here we're looking at linear mixed effects models with random intercepts and slopes, evaluating the association between average PM2.5 exposure during disease and the rate of decline in forced vital capacity. And what you can see on the right here is a figure facetted by PM2.5 during disease quartile, and the lung function trajectories of each patient in that quartile represented by each line. And the overlap of that is a smooth blue line that represents the average trend of lung function in each of these quartiles. And so in the linear mixed models, we elected to use a random intercept and a random slope because this essentially says that each patient can experience a different starting point, i.e. intercept, and a different slope, i.e. rate of decline in their lung function. And this is consistent with what we know of the behavior of fibrotic ILDs in that not all patients progress at the same rate or in the same manner. So knowing all of this background, we can see that in these models, adjusted for the same covariates as I mentioned before, that patients in quartile 4 experience a significant interaction with time with a beta of negative 1.54. And this can be interpreted as, compared to quartile 1, patients in quartile 4 of PM2.5 exposure during disease experience an additional 1.54 unit decrease in percent predicted FVC per year. The continuous PM2.5 during disease exposure variable was also significant in these models with a beta of negative 0.25, indicating that for each 1 microgram per meter cubed increase in PM2.5 during the course of disease, patients experience an additional 0.25 unit decline in FVC percent predicted per year. Next, we performed the same analysis, instead looking at the time period of 5 years prior to censoring. And we can see that in these models adjusted for the same covariates, that patients in quartile 2 to quartile 4 all experience a significant interaction with time compared to quartile 1. And again, the beta for quartile 4 is negative 3.16, indicating that compared to quartile 1, patients in quartile 4 experience an additional 3.16 unit decrease in percent predicted FVC per year. And the continuous PM2.5 exposure in the 5 years prior to censoring was also significant here. We also performed these analyses for diffusion capacity for carbon monoxide, adjusted for the same covariates. And we found that patients in quartile 4 experienced a significant interaction with time compared to quartile 1 with a beta of negative 1.16, indicating that compared to quartile 1, patients in quartile 4 of PM2.5 exposure during disease experienced an additional 1.16 unit decrease in percent predicted DLCO per year, but our continuous exposure variable did not meet statistical significance. We performed the same analysis, again, now looking at the time period of 5 years prior to censoring, and we can see that quartiles 3 and quartiles 4 are significant compared to quartile 1. And in these models with this time period, the continuous PM2.5 exposure in the 5 years prior to censoring had a significant interaction with time. The beta of negative 0.31 can be interpreted as for each 1 microgram per meter cubed increase in PM2.5, there is an additional 0.31 unit decrease in DLCO percent predicted per year. So to summarize the lung function results, patients that experience higher PM2.5 exposure in the 5 years pre-diagnosis have lower baseline FVC, but no difference in baseline DLCO. Patients with higher PM2.5 exposure during disease and in the 5 years pre-censoring experience both a more rapid decline in FVC and a more rapid decline in DLCO. And this is the first data to link air pollution exposure with baseline lung function differences or with differences in the rate of decline of DLCO, which is quite novel, and also the first to look at these outcomes in patients with a diverse cohort of fibrotic ILDs other than just IPF. So moving on from the impact of air pollution on clinical outcomes in patients with fibrotic ILD, I briefly wanted to introduce the next steps of my PhD research. So the other major question I had was, what role does epigenetics play in ILD, and how do these epigenetic factors respond to air pollution exposure? I'll just provide a little brief background on this with some information about the methodological approach I plan to take to address these questions and some very preliminary work. So we know that there are both peripheral blood and lung tissue changes in DNA methylation that are seen in patients with IPF, which is the most common form of ILD in comparison to controls. And this is a study that my primary mentor, Dr. Yingzi Zhang, collaborated on, which demonstrated that there were around 147 differentially methylated regions in patients with IPF compared to controls. And a number of these differentially methylated regions were associated with genetic loci of relevance to ILD pathophysiology, as shown in panel D here. They also found that these regions were associated with downstream gene expression changes. Other research that has been performed in non-diseased individuals has shown that exposure to pollutants like nitrogen dioxide is associated with altered DNA methylation patterns, as shown in this table here, and that these alterations may actually mediate some of the association between air pollution exposure and reduced lung function. So I plan on evaluating this in our cohort of patients with fibrotic ILD. And furthermore, line 1 DNA methylation, which is a surrogate measure of global DNA methylation at long interspersed nuclear elements across the genome, is also associated with PM10 exposure, and this was shown in a healthy cohort of Italian women. So the last aim of my PhD project will look at the impact of air pollution exposures on DNA methylation and gene expression patterns in peripheral blood samples from a cohort of patients with fibrotic ILD. Peripheral blood DNA methylation levels have been shown to be associated with inhaled exposures, including smoking and air pollution exposure. So we believe that in this patient population, that it may serve as a valuable biomarker of historical environmental exposures. We will use two approaches to evaluate DNA methylation patterns in patients with fibrotic ILD, beginning with a global DNA methylation analysis using long interspersed nuclear element or line 1 based assay. And then we'll use a chip-based assay to look at air pollution impacts on epigenome-wide DNA methylation patterns. Next, we will look at the impacts of air pollution exposure on selected genetic loci that are highly associated with the phenotype of IPF based on GWAS studies in this population, and which are also highly expressed in peripheral blood. And lastly, we'll use some previously performed peripheral blood mononuclear cell gene expression analyses from patients with IPF to look for associations between gene expression patterns and air pollution exposure. As I mentioned in the previous slide, we're using two approaches to evaluate DNA methylation in peripheral blood samples. The first is using line 1 DNA methylation, and line 1 represents 17 to 18% of the genome and has been shown to serve as a useful surrogate measure of global DNA methylation. We'll perform this assay on a sample of patients from the PFF registry, since we have access to the largest number of samples from this cohort and the most diverse geographic distribution, which spans across multiple ILD care centers in the US. Our funding from the PFF will allow us to perform this line 1 DNA methylation on approximately 1,000 samples from PFF and another 100 from Simmons Center. And subsequently, we'll perform the more granular Infinium methylation epic 850K bead chip array, which is an epigenome-wide method of DNA methylation analysis that will allow us to interrogate differentially methylated loci, which we're unable to do with the line 1 DNA methylation analysis. To finish up, I briefly wanted to go over what our next steps are. So first, I need to confirm the clinical outcome results I've shown you in the Simmons cohort with the PFF patient registry cohort and the Canadian cohort from CarePF. I'll then add to the complexity of my models by performing multi-pollutant analyses to consider the intersecting impacts of other pollutants on one another. I'm currently in the process of acquiring PFF samples to perform the line 1 DNA methylation analysis, which I hope to perform by the end of the year. And moving forward, I'll likely, upon completion of my PhD, perform multiple studies investigating the impact of air pollution on other epigenetic mechanisms such as histone modifications and non-coding RNAs, as shown in the mechanistic figure here on the right that's taken from our 2020 Blue Journal Review manuscript. And most importantly, I'm going to continue to advocate for better air pollution policies to protect patients with ILD. I'm currently a member of the Environmental Health Policy Committee with the American Thoracic Society, and through that avenue and my participation with PFF, I will continue advocating for that goal. And of course, I couldn't have done any of this without the incredible support of my many mentors and collaborators at multiple institutions, as well as the funding support I've received. Thank you.
Good afternoon. My name is Bridget Graney. I am an assistant professor of medicine at Denver Health Medical Center and the University of Colorado Anschutz Medical Campus. I'm the co-director of the Interstitial Lung Disease Program at the university, and I am one of the 2020 Pulmonary Fibrosis Foundation Scholars. And I'll be speaking to you about my research today that's focused on improving patient and caregiver outcomes in pulmonary fibrosis by specifically focusing on caregiver burden. I first want to say thank you to my mentorship team, specifically Dr. Joyce Lee and Dr. David Beckelman at the university, our entire interstitial lung disease program at the university, and of course, a special thank you to the Pulmonary Fibrosis Foundation for funding this work and for inviting me to speak about it today. I focus on a specific area of clinical research in patient-reported outcomes research. Patient-reported outcomes research is focused on capturing patients' experience of disease in a quantifiable way so that we can further understand their lived experience. Through this, we use validated questionnaires, qualitative studies, and other measures to try to quantify these perceptions of health status, symptoms, and quality of life. These are particularly important for individuals living with incurable diseases so that we can identify areas for intervention that may be modifiable outside of their disease process. We are also increasingly using patient-reported outcomes to identify efficacy of therapeutics that are incorporated into the FDA approval process. I first began this work with Jeff Swigris at National Jewish, where we were looking at patient and caregiver experiences with the use of supplemental oxygen. And what we found that is in addition to the life changes that patients experience when they start using oxygen, caregivers' lives also changed significantly. Caregivers had to adapt their lives to oxygen, accept the life changes that they had as a result of oxygen, and also see the impact of oxygen therapy on the individuals they're caring for. One caregiver told us that everything changes with oxygen. You have to work everything in your life around it, and there are certain things you just can't do. It's changed life a lot, but definitely not for the better. This led us to describe the shrinking world of oxygen therapy, where patients and caregivers alike struggle with not being able to live their lives as they did before oxygen therapy. Because of this understanding that caregivers are also impacted by the disease that their loved ones lived with, I became very focused on trying to understand caregivers' experiences in the world of pulmonary fibrosis, in addition to patients. This has led to my long-term goal, which is to reduce symptoms and improve quality of life for individuals with pulmonary fibrosis, as well as their caregivers, by developing and implementing dyadic interventions. With that goal in mind, I'd like to tell you more about my research. So today we're going to discuss the importance of family or informal caregivers, the impact of caregiving on caregiver and patient health, our current understanding of caregiver burden in pulmonary fibrosis, and to talk about my research project evaluating caregiver burden in a dyadic mixed method study. For the purposes of this talk, these are the definitions we're going to use. A patient is any individual diagnosed and living with pulmonary fibrosis. And the term caregiver, although fraught and not all-encompassing, and there may be better words, is going to be used to describe a person close to an individual living with an illness, such as a family member or friend who provides unpaid care or support to the individual. Other terms that are frequently used to describe caregivers are family, informal, or unpaid caregivers, but I'll be using caregivers today. So first, we're going to discuss the importance of caregivers and the impact of caregiving on caregiver health. Caregiving encompasses many different things. It can include helping with activities of daily living, such as bathing, showering, and getting dressed, to helping with housework, meal prep, finances, medications, and so on. Caregiving also includes communication, coordination of medical care, and advocacy on behalf of the patient loved one. Caregivers also do a lot of monitoring for their patients to ensure that they're doing well, and they make care plan adjustments based on their status. Some caregiving is specifically to assist with skilled medical or nursing tasks, and that can be included as well, such as changing ostomy bags, urinary catheters, etc. Importantly, people do not have to live in the same home or be related to the person to be a caregiver. Caregiving in the United States is highly prevalent, and it occurs across all generations, racial and ethnic groups, income and educational levels, etc. Nearly 42 million Americans provide unpaid care to an adult over the age of 50 in the past year, where they spend an average of 24 hours per week on caregiving activities, with spousal caregivers providing more hours than non-spousal caregivers. If you aggregate this amount of work, it's estimated that there's $470 billion of unpaid care that is related to this informal caregiving in the United States. The caregiver experience is highly individualized. Some caregivers experience a significant amount of positive and rewarding aspects to caregiving, with the majority saying that they feel a sense of purpose and meaning in their life as a result of caregiving. However, some caregivers do experience negative impacts as a result of caregiving. These negative impacts are termed caregiver burden, which is the extent to which caregivers perceive that caregiving has an adverse impact on their emotional, social, financial, physical, and spiritual functioning or well-being. Caregiver burden is also highly prevalent, with 60% of caregivers experiencing moderate to high burden. These estimates are based on what's called the level of care index, which incorporates hours of care and how much time is spent both on activities of daily living, such as bathing and dressing, and instrumental activities of daily living, such as finances, grocery shopping, etc. But this is a significant number of caregivers who experience high to moderate burden. The risk factors for the development of caregiver burden include living with the care recipient, providing more hours of care, the degree of financial stress that caregiving leads to, for example, if people have to leave a job to be a caregiver, being a female caregiver, and then caring for individuals who have significant impairments in their functional capacity. Because of the impacts of caregiver burden, many caregivers have health-related impacts as well. 21% of caregivers describe their health as being fair or poor, which is significantly higher than the general population. Nearly a quarter of caregivers feel that caregiving in and of itself has made their health worse, and 36% report high emotional stress as a result of caregiving. Caregivers who report lower self-rated health or that they have had health impacts have the same risk factors for poor health as they do with caregiver burden. Again, because caregiver burden is this all-encompassing term of the impact of caregiving. Because of the health impacts that caregivers experience, they're often described as invisible second patients due to the health impacts that they experience. The physical symptoms related to caregiving are independently associated with mortality. There is a 63% greater risk for mortality in caregivers who experience high levels of caregiver strain or burden, and those with lower strain have improvements in mortality. There's also significant psychological disease in individuals who provide care, including depression, anxiety, and suicide. What's interesting is that even caregiver status is also related to patient outcomes. Caregivers with higher burden see that their patient loved ones has increased healthcare utilization, and caregivers with less burden care for patients who have higher confidence and maintenance of self-care. Although this is likely related to the functional status of the care recipient, the fact that caregiver burden is related to patient outcomes as well is important. We also know that the psychological health of caregivers and their patient loved ones is directly related. There is a correlation between both depression and anxiety between patients and caregivers. This led us to ask, do caregivers of individuals with pulmonary fibrosis experience significant care burden, given that patients living with pulmonary fibrosis experience high symptom burden, impaired functional status, and impairments in quality of life? And if caregivers do experience high levels of burden, can we design interventions that will decrease burden and, given the relationship between patient and caregiver outcomes, improve outcomes for both individuals? First, we must review what we know about caregiver burden in pulmonary fibrosis. To my knowledge, there are currently 15 studies that include or specifically assess caregivers in the pulmonary fibrosis literature. None of these studies specifically measure caregiver burden, but many are qualitative studies that describe the lived experiences of caregivers of patients with pulmonary fibrosis. And they suggest that they likely do experience significant burden given the impacts on social, emotional, and physical well-being. But what can we learn from other disease states? So in the oncology literature, dementia literature, heart disease, and to some extent COPD literature, caregivers have been evaluated more rigorously. Because patients living with dementia often have different caregiver needs, I'm going to focus specifically on insights from the oncology world. Within the oncology world, there is wide recognition of the importance of family caregivers. They can be directly involved in the delivery of complex interventions, participate in shared decision-making, and of course, function as key advocates and care coordinators. There is a large number of caregiver-focused studies, but a relatively small number that focus on patient-caregiver pairs, otherwise known as dyads. There are, of the small number of dyadic interventions, they have been shown to demonstrate improvements in quality of life, relationship status, as well as depression and anxiety. To summarize, caregivers are a crucially important part of our social fabric in the United States. And many caregivers experience caregiver burden as a result of caregiving. High levels of caregiver burden are independently associated with worse health outcomes, including mortality, to the point where caregivers are often described as the invisible second patients. Knowing that caregivers and patients certainly don't exist in isolation, there is an interdependence to these outcomes, both positive outcomes and negative outcomes. And the qualitative research we have to date suggests that caregivers of patients with pulmonary fibrosis likely experience caregiver burden. But as yet, it has not been systematically evaluated to understand the prevalence, severity, and drivers of burden in this population, which takes me to my research, describing our approach to evaluating caregiver burden in a dyadic mixed methods study. Again, recognizing that caregivers likely experience burden and that highly burdened caregivers have increased rates of adverse health outcomes, our long-term goal is to reduce caregiver burden to improve outcomes for both individuals, including symptoms, overall health, and quality of life. To do that, we first must understand caregiver burden in pulmonary fibrosis. Our central hypotheses are that caregivers of patients with pulmonary fibrosis experience high levels of caregiver burden that are associated with patient and caregiver-specific factors, including disease severity, symptom burden, quality of life, and resiliency. Caregiver burden is complex, and it is influenced by factors that are intrinsic and extrinsic to the patient and caregiver. To begin to study this, I adapted the Perlin stress process model of caregiving as the conceptual framework for this work that describes the background and context in which patients and caregivers live, stressors, and moderating factors that help to either relieve stress or can increase stress that lead to outcomes for both individuals. Within our conceptual framework, the background and context include caregiver self-reported health, the care demands that the caregiver faces, and the stressors can include things like patient disease severity and other patient illness-related factors. Secondary stressors are sometimes described as the spillover effects of caregiving in the context in which care is provided. That can include relationship satisfaction, changes in lifestyle, work, schedules, and so on. There are many moderating factors, including caregiver cognitive and behavior responses, coping mechanisms, etc. For this particular study, we're going to focus on resilience as a potential moderating factor for these stressors. And outcomes include both patient and caregiver outcomes, including caregiver burden, quality of life, psychological health, and the benefits that caregivers find as a result of caregiving. To attempt to answer these questions, I'm using an explanatory sequential mixed methods approach with 3 phases: a quantitative, qualitative, and an integrated interpretation of this data. The study participants are patients diagnosed with pulmonary fibrosis based on multidisciplinary diagnosis and their self-reported caregivers. We are targeting an enrollment of 50 dyads, so 100 total individuals in patient-caregiver pairs, so 1 patient and 1 caregiver enrolled together. The age of the patient is going to be between 40 and 89 years old for inclusion, and the caregiver can be any adult that is identified as the caregiver. For phase 1, we will use quantitative data collection with questionnaires that align with the 4 domains of the stress process model, again, including background, stressors, mediators, and outcomes. You can see the caregiver questionnaires and the patient questionnaires on this slide. These are all valid, reliable measures that have been used in prior research. And for the caregiver questionnaires, we're focused on, of course, measuring caregiver burden, measuring impacts such as caregiver benefits. Moderators for this study will be resilience, and then understanding quality of life, psychological health with screening for depression, as well as the relationship with the care recipient. Patient questionnaires are similar, including quality of life measurements, but we are including the King's Brief ILD questionnaire for disease-specific quality of life. And then the other items remain the same. Additional data we'll be collecting include baseline demographics, as well as patient medical data, including the specific ILD diagnosis from a multidisciplinary discussion, the most recent pulmonary function testing for an estimate of disease severity, other medications, comorbidities, and supplemental oxygen prescription. In the second phase, we will collect qualitative data for collection and analysis. To perform the qualitative analysis, we will first integrate the results that we find in phase 1 into the development of a semi-structured interview guide. The reason for this is the currently existing measurements for caregiver burden are really focused on caregiving tasks and burden issues that are primarily found in the dementia world, meaning a significant focus on helping individuals with activities of daily living. So we will use the results from the quantitative portion to inform the development of the interview guide to try to understand the drivers of caregiver burden that are specific to patients and caregivers with pulmonary fibrosis. We're going to use a purposeful stratified sampling approach where we identify dyads to complete these interviews. We will use the top 50% of dyads with the highest reported caregiver burden to participate in the interviews. Of course, inviting sequential dyads if individuals are unwilling to participate in the interview process. We are planning to interview both the patient and the caregiver of each dyad in separate interviews. This will allow both individuals to honestly describe their experiences about caregiver burden, as well as to have the patient perspective on how caregiving may impact the caregiver in a way that the caregiver is not always as aware of. In phase 3, we will integrate the data from the quantitative and the qualitative studies for complete interpretation of caregiver burden in this population. To do that, we will integrate the quantitative and qualitative data. The quantitative data will have already been evaluated with summary statistics generated. We will also do an exploratory actor-partner independence modeling, which is a way to understand how one individual's current behavior or status influences another individual's current behavior or status. This is exploratory in nature for this study because of our sample size, but we thought that this was an important thing as we begin to explore dyadic relationships and how one individual impacts the other. This is a way to incorporate interdependence in outcomes, and for this study specifically looking at patient-related health status, caregiver burden, and reported quality of life. For the qualitative data, we will use a grounded theory approach and general inductive coding in our analysis, and integrating that again with the results of the quantitative data to try to fully describe the experience of caregiver burden in this population. This work is important because it evaluates in a rigorous manner caregiver burden in this population, which has not previously been described. I am utilizing a mixed methods approach, which will be a robust way to understand both quantitatively and qualitatively the experience of caregivers in pulmonary fibrosis. Studying dyads together will also enrich this data, having both the patient experience as well as the caregiver experience. We are also doing a preliminary evaluation of a moderating factor, specifically resilience, to see if we can identify a potential target for future interventions. We know that resiliency is both a teachable and modifiable trait and has been shown to be associated with caregiver burden in other disease states, which is why we chose this to evaluate. In summary, caregiver burden is prevalent and is associated with worsened health outcomes in affected caregivers and patients. Our current literature suggests that caregivers of patients with pulmonary fibrosis experience burden, but its extent and subsequent impacts have not yet been quantified. I will utilize a novel dyadic mixed methods approach to further our understanding of caregiver burden in pulmonary fibrosis to meet the long-term goal of lessening caregiver burden and improving quality of life for both patients and their caregivers. And I want to thank you very much for inviting me to speak today. I'd be happy to answer any questions.
Hello, I'm Rami Unterman from Tel Aviv Medical Center. I wish to thank the PFF for the opportunity to present our research, which focuses on the peripheral blood in IPF and other fibrotic lung diseases using single-cell profiling techniques. These are my disclosures. This work was mostly done while I was the research fellow at Naftali Kaminsky's lab, and this ongoing work is partly supported by my PFF Scholars Award. This is the outline of my talk. First, we're going to start with the rationale, why focus on peripheral blood in IPF anyways. Then a short introduction to single-cell RNA sequencing. Then we're going to present our research regarding single-cell immune profiling in IPF, including our lung blood recruitment model in IPF. We're going to present also some preliminary data on single-cell profiling in IPF versus HP, hypersensitivity pneumonitis, and then conclusion and future directions. So why focus on the peripheral blood in IPF? First, blood is an accessible window to explore immune changes in IPF. Previous reports showed strong prognostic signals in the blood, including some plasma proteins like MMP7 and CCL18, monocytes, and the 52 gene signature, which we're going to mention shortly. Identifying distinct immune aberrations in IPF and other ILDs could lead to more accurate diagnosis and potential targeted therapy based on a patient's immune profile. Let's look at some of the evidence for the prognostic importance of peripheral blood in IPF. This figure is taken from a paper by Jose Razomaya and Naftali Kaminsky describing an outcome predictive 52 gene signature in peripheral blood, which was validated in 6 different cohorts. As you can see in the figure, the Kaplan-Meier curves separate high-risk and low-risk patients based on their peripheral blood transcriptome. And more recently, 2 papers, one by Madeleine Scott et al., and a second one by Michael Kreuter, Toby Maher, and colleagues, established the prognostic importance of increased blood monocytes as markers of worse outcome. Based on these data, we felt that deep profiling of the peripheral blood in IPF using single-cell RNA sequencing technology is warranted. In the next slides, I'm going to present our research entitled Single-Cell Profiling Reveals Immune Aberrations in IPF Associated with Disease Progression. But first, a brief introduction to what single-cell RNA sequencing is. So let's assume we've got a tissue, for example, lung tissue. This tissue is comprised of many cell types. So if we're going to run bulk RNA sequencing, we're going to take this tissue, we're going to grind it, we're going to extract the RNA, and then sequencing it to get the average gene expression from all cells. Cellular heterogeneity is, of course, masked. And recently, in the past few years, we've got the single-cell RNA sequencing technology. In single-cell RNA sequencing, we first dissociate the tissue into single cells and then sequence every single cell individually using a barcoding system. This allows us to separate different cell types according to the gene expression profile and determine the gene expression profile for each and every cell subpopulation separately. In recent years, single-cell RNA sequencing led to many new discoveries, including the ionocytes in the array epithelium, new types of dendritic cells and monocytes, and recently the aberrant basaloid cells in the IPF lung. So our objectives for this study were to identify shifts in blood immune cell populations and phenotypes that are reflective of the presence or progression of IPF, to determine the cellular source of the outcome predictive 52 gene signature, and to use our blood and lung single cell atlases to construct a lung blood immune recruitment model in IPF and validate it. So this is our methodology. We used cryopreserved PBMCs that were processed using the 10x chromium 5' single cell platform. 38 subjects in 3 matched subgroups were included, 12 progressive IPF patients, 13 stable IPF patients, and 13 matched controls. IPF subjects were all untreated at the time of blood draw. All subgroups were matched for age, gender, and smoking status, and progressive and stable IPF subgroups were also matched for pulmonary function tests. The difference between stable and progressive IPF patients was whether or not they died within a 3 years follow-up. And you can see here the survival curve of progressive IPF versus stable IPF. This is the overall study design in which we investigate both the blood and the lungs. It includes 5' single-cell RNA sequencing of PBMCs using a 10x platform, reanalysis of lung single-cell RNA data focusing on the immune system, as well as protein marker validations from both plasma samples and lung tissue homogenates. The last step is data integration and creation of a lung blood recruitment model based on our findings. Our data set included 153,000 cells from 38 subjects. In this plot, which is called a UMAP, every single dot is a cell. Every cell has thousands of genes that are detected. These cells were clustered based on their gene expression profile, and 23 distinct cell types were identified. Cell type detection was enhanced by our 5' methodology. We noticed significant shifts in immune populations between conditions. While monocytes, as expected from prior studies, gradually increased between control, stable, and progressive IPF, as you can see on the left box plot. Lymphocytes gradually decreased, as you can see in the right. Next, we drill down to the subpopulation level. You can see that classical and intermediate monocytes increase in IPF, while non-classical monocytes are not increased. See all the monocyte subpopulations in the green rectangle. What about lymphocytes? Naive CD4 T cells, naive CD8 T cells, and effector T cells decrease in progressive IPF. And while the general trend of most lymphocytes is to decrease in progressive IPF, we noticed that T regulatory cells, or T-Regs, show the opposite trend of increasing in progressive IPF patients, as you can see in the blue rectangle. And we'll elaborate on this in the upcoming slides. What about the 52 gene signature? In this dot plot, you can see the cell types on the y-axis, the 52 genes that were associated with prognosis on the x-axis. Each dot, its color intensity represents the average expression of this gene in the specific cell type, while the size of the dot represents the percentage of cells expressing this gene. As you can see, all the genes that are increased in association with worse prognosis, all the 7 genes that are increased with worse outcome are of monocyte origin. That led us to believe that, at least in part, this 52 gene signature is driven by shifts in cell population, the increase in monocytes, the decrease in lymphocytes that we observed. Regulatory T cells, or T-Regs, are increased in progressive disease. And this is one of our most important findings. You can see in this slide, there's a statistically significant increase in progressive versus stable IPF. Moreover, when we plot the survival curves according to whether or not patient had high T regulatory cells or low T regulatory cells, we see that these curves separate nicely when the high T-Regs patients have worse prognosis. And this had almost a statistically significant result. It was a strong trend. We believe that our small sample size was the reason that it didn't reach statistically significant level. But that's not all. T-Regs are also increased in the IPF lung. And here you can see the results from 2 different lung single-cell RNA-seq datasets, one from Kaminsky and Rosas, and the second one from Kropsky and Banovich. As you can see in these box plots, the level of T-Regs in IPF versus control in the lung was increased in both datasets. If we look into specific pathways in T-Regs, we can see that the TGF-beta SMAD, or mTOR, and the IL-2 pathways are increased in progressive versus stable IPF, while the interferon gamma pathway, which is an inflammatory pathway, is decreased. And as you can see, TGF-beta and mTOR and IL-2 pathways are activation pathways in T-Regs. So T-Regs seem more active and not only increased in numbers in progressive IPF. So what do we know about T-Regs in IPF? The role of T-Regs in IPF has previously been studied, yielding conflicting results. Our study found increased T-Regs in progressive IPF and decreased in stable IPF. So if you look again on the box plot, right corner, you can see that there is statistically significant decrease in T-Reg numbers in stable IPF versus controls, and then statistically significant increase in progressive versus stable IPF. Most previous studies support our findings. However, Kotsianidis et al. Reported reduced quantities and function of T-Regs in peripheral blood of patients with IPF compared to healthy controls. However, they did not separate IPF patients according to severity, and this might explain this discrepancy because, as we showed, there's a big difference between stable and IPF patients with regard to T-Regs. Fibrocytes, called Gen 1A1 positive, CD45 positive cells were rarely found and did not form their own clusters. So next, we ask ourselves, can gene expression of specific blood cells distinguish stable from progressive IPF? As you can see in this heat map, stable IPF patients are to the left and progressive IPF patients are to the right. And each column is a patient, each row is a gene. We'll focus on the upper part, which is the monocytes. TNF alpha and IL-1 beta pathways were increased in monocytes of stable IPF patients compared to progressive ones, suggesting that at least in some stable patients, there is a pro-inflammatory state in monocytes. If we look at IL-1 beta for an example, we can see the higher expression in stable patient in orange and the low expression in progressive patients. However, as you can see, there is significant inter-subject variability. Next, we ask ourselves, are there any cytokines that may play a role in monocyte and Treg increases in IPF? In order to answer this question, we decided to measure several proteins in the serum samples of our patients. Colony-stimulating factors, including MCSF, GCSF, and GM-CSF. Monocyte chemoattractant proteins, or MCPs, including CCL2, CCL7, CCL8, CCL13, which are all ligands of CCR2 that, as you can see to the right, are expressed mainly on classical monocytes and are believed to be the chemokine receptors of classical monocytes, as well as Treg chemoattractants, CCL17, CCL22 that are ligands of CCR4, CCL1, CCL18 that are ligands of CCR8. And those two receptors, CCR4 and CCR8, are specific receptors on Tregs that are likely involved in Treg chemoattraction. So I'm going to show you positive results. So for classical monocytes, MCP3, CCL7 was increased in the plasma in both progressive and stable IPF. CCL13 or MCP4, while it was not increased in our plasma samples, it was significantly increased in the lung tissue homogenates of IPF patients versus controls. Treg chemoattractants, so CCL22 was increased in the plasma of IPF patients versus controls and significantly increased in lung tissue homogenates of IPF versus controls. CCL18, a very known prognostic biomarkers in IPF, was increased in the plasma of IPF patients versus controls. So these three proteins, CCL7, CCL18, CCL22, are likely candidates to be involved in chemoattraction of classical monocytes and Tregs, respectively, from the blood into the IPF lung. So we were intrigued about the role of CCL22 in IPF. So CCL22, CCR4 ligand is increased both in IPF plasma and IPF lung tissue homogenates, as we showed, which makes it a likely candidate for Treg chemoattractions in IPF. So CCL22 was previously found to be secreted from lung tissue and to selectively recruit Tregs into the lungs. And in another study, CCL22 was increased in bowel fluid and its levels inversely correlated with the diffusion capacity. Next, we were interested in mapping the origin of CCL22 in the lung. And here you can see data from the single cell RNA sequencing datasets of the lung from two datasets, Kaminsky-Rosas and Kropsky-Banovich. And you can, as you can see, CCL22 originates from dendritic cells. So we did the same in order to map the origin of all other proteins. And then we devised a proposed lung blood recruitment model in IPF. So here you can see a blood vessel in which we have T-Reg and classical monocyte with their respective receptors. This, on the right, you can see the lung compartment with T-Reg, dendritic cells, macrophage, and myofibroblast. The dendritic cell expresses and secretes CCL22, which acts on CCR4 receptors on T-Regs. And the macrophage secretes CCL18, which acts on CCR8 on T-Regs, leading to recruitment of T-Regs from the blood into the IPF lung. Macrophage also secretes CCL7, acting on the CCR2 receptor. And both the macrophage and the myofibroblast secrete CCL13. Both of these agents are likely candidates to act as monocyte chemoattractants from the blood into the IPF lung. And as we all know, classical monocytes are considered to be the precursors of macrophages in the lung. T-Regs are known to induce an alternative activation pathway in macrophages with TGF-beta and IL-10, and in turn, macrophages secrete profibrotic mediators that act on myofibroblasts. So in summary, we provide a first single-cell atlas of the peripheral immune system in IPF according to disease progression. Monocytes increase in progressive disease, lymphocytes decrease. Monocytes have less inflammatory phenotype in progressive IPF and are predictive of increased mortality in IPF. T-Regs are activated and increased in progressive IPF in both the blood and the lung and may be predictive of increased mortality. And the outcome predictive 52 gene signature is driven, at least in part, by shifts in PBMC composition. CCL7 for classical monocytes and CCL18 and 22 for T-Regs are likely chemoattractants of immune cells into the IPF lung. Based on our findings, we propose a lung blood recruitment model in IPF. What about other fibrotic lung diseases such as hypersensitivity pneumonitis? These are preliminary results from work that's mostly done by Amy Zhao, an MD PhD candidate at the Kaminsky lab. So we started with 16 samples, 6 chronic hypersensitivity pneumonitis, 5 IPF, and 5 controls. These were subjected both to gene expression profiling and T-cell and B-cell receptor repertoire analysis, which is another benefit of the 5' methodology. This is the integrated UMAP of these 16 samples together with the previous 38 samples of IPF patients at control, yielding about 220,000 cells. And all cell types were detected in our data set. So we asked ourselves 2 questions. Can cell composition differentiate CHP from IPF? And can gene expression distinguish CHP from IPF? CHP patients have significantly higher lymphocyte percentage than IPF. And although we only had few CHP patients, it was statistically significant, indicating a high effect size. This was true both for all T lymphocytes and for CD4 T lymphocytes specifically. And can gene expression distinguish CHP from IPF? So CHP cells show differentially expressed gene enrichment in pro-inflammatory pathways, including the interferon gamma response and the TNF alpha signaling via the NF kappa beta. Then we conducted T-cell reclustering, which revealed a CHP-enriched cluster, as you can see to the left. It's marked in green, and you can see it's highly enriched for CHP cells. This CHP-enriched T-cell cluster was found to be enriched with IL-12 signaling pathway and IL-2 signaling pathway, which may indicate it's an activated CD8 T-cell cluster. What about T-cell receptor repertoire analysis? So this is very preliminary results, but we can see here that there is a trend towards reduced T-cell clonality in CHP samples compared to IPF and controls. So in summary, T lymphocytes, and specifically CD4 T lymphocytes, are highly enriched in CHP versus IPF. CD8 T lymphocytes display a pro-inflammatory activated phenotype, and T-cell receptor repertoire exhibits a trend towards reduced clonality in CHP samples. In conclusion, major shifts in PBMC subpopulations occur in IPF and HP and are of prognostic value. T-Regs are activated and increased in progressive IPF in both the blood and the lung and may be predictive of increased mortality. Specific chemokines are likely involved in the migration of immune cells from the blood into the IPF lung. And our future directions are extending our PBMC single-cell RNA sequencing research to other fibrotic lung diseases, exploring the role of the identified chemokines in animal models of PF, gaining deeper insight into the T-cell receptor and B-cell receptor repertoires in lung fibrosis. And with that, I wish to thank the PFF for funding this research and our great team at the Kaminsky Lab and the LPCCSM, as well as our other external collaborators. And this photo from the PFF Summit 2019 is a reminder of how much fun it was to meet in person. And I hope we can all meet again in the next PFF Summit. Thank you.
Welcome back. I'm Mike Beers. I have been the chair for this session, and it has been a wonderful session for me because, as I said prior, I had served as the chair for the respiratory review committee here for all of these grants. I saw these proposals coming through in 2019, 2020, and it's really great to see all of you have progressed in what you are doing as you go forward. And I think the PFF should be proud of its choices in funding your work. So with that, we have some questions in the chat. We've got some questions in my unfiltered sheet down here. I'm going to combine a couple here for Dr. Unterman first. And the question is, what do you think the T-Regs are actually doing in IPF? So, it is controversial as to, their many functions. And so, if you could comment a little bit on that to get us going.
So that's a great question. Thank you for that. So the truth is that we don't really know. But since T-Regs are immunomodulatory cells, they tend to be anti-inflammatory, decrease the flames of inflammation in the tissues. So one can speculate that they have an anti-inflammatory role. We know that the immune system is involved in IPF. It's not the driver in IPF, but it is involved. But we don't really know exactly what's the role of T-Regs. I think there's plenty of indication to consider T-Regs as a player in IPF. There's much more work to be done in order to elucidate that fully.
Okay, good. So Dr. Gooby, full disclosure, my wife is a is the Director of Enforcement for the Environmental Protection Agency. So your work is near and dear to my heart. And I also was involved with our Center for Environmental Toxicology here at Penn. And so I was going to ask you, a question related to this whole issue of how do you validate the exposome? So the choice for your study was to use satellite data. And this has been done also to try to figure out what's going on in the burn pits in Iraq and try to use the sort of similar data, right? So the question is, has that data been validated with wearables or some other way to really say that if I live in this zip code, this 1 center, km squared zip code, then I have this exposure, which I think is critical to long-term work that you're doing.
That's a fabulous question, Dr. Beers. So it's actually interesting with these satellite-derived hybrid models. They do something called a 10-fold cross-validation approach with ground-based EPA monitoring data. And so they'll actually take PM2.5 measurements at these ground-based sources, and they'll predict what they expect that measurement based on their satellite-derived models and their atmospheric composition analyses and other ground-based measurements outside of that to predict what they expect the measurement to be at that ground-based monitor. And they do that in a 10-fold cross-validation approach that does provide quite a bit of accuracy. In terms of the validation to wearable monitors, I'm not sure that that's being performed with these data sets. Certainly, these types of approaches can't capture the day-to-day variability that someone would have in moving from their home to potentially occupational site, or we're talking about younger populations going to school and other areas that they might be traveling to throughout their day-to-day life. So there is going to be that inherent variability that we're not able to capture with these longer-term exposure studies. But I do think that the spatial accuracy that we get from these satellite hybrid models is much better than many other types of exposure estimation techniques.
And can you distinguish the exposome from all of the other factors that might be involved? Like, people that live in, challenged neighborhoods have a lot going on, including access to care and other issues. And so the question is how do you sort this out from that?
Another fabulous question. We're actually, we just had a paper accepted for publication that's looking at the impact of neighborhood level disadvantage on patients with fibrotic ILD in this cohort. It hasn't been released yet, but just found out a couple of days ago that it got accepted. And so we incorporate that assessment of neighborhood level disadvantage into our pollution analyses as a covariate to help control for the socioeconomic and sociodemographic factors, given that these factors can also be associated with higher pollution exposures throughout the US. We are limited in our ability to capture occupational exposure history in all of the cohorts that we're evaluating. So certainly there is probably that component, as well as the indoor exposome component that we're not necessarily able to capture, that might be better evaluated in smaller studies that are measuring personal exposures to indoor pollutants.
I want to drag Dr. Graney into this because we're talking about socioeconomic status and in terms of burden. You mentioned financial stress, but I would imagine that financial stress is really a correlate of socioeconomic status in terms of burden on caregivers. Is that, what do you think?
I think that's probably part of the answer. I think it's more complicated than that, particularly for caregivers. What that doesn't, the financial burden piece on a broader level takes into account the fact that many caregivers have to leave work or change their work, conversely, to pay for the cost of care. Some caregivers, in addition to their caregiving duties, actually have to take on more work outside the home to either have insurance through employer-based insurance programs or to be able to afford the cost of care. So the financial burden piece becomes really challenging to tease out and understand because of the many different ways it can change depending on caregiving needs.
Yeah, that's what I would have suspected. Okay. So Dr. Unterman, there's a question in the chat. Did you see any changes in CCL17 in this?
Yeah, that's also a very good question. So the reason I didn't mention CCL17 was because it wasn't increased in our plasma samples. However, there is a lot of data showing that it is increased in IPF, and I think it may have also a role as a chemoattractant for T-Regs along with the CCL22.
Right. Yeah. And then I guess to follow up on other sort of performance characteristics of your test. So have you looked at this sequentially? So this gene signature says, if I take this as a snapshot in time, it predicts progression to, of IPF. But have you actually been able to follow this sort of, how does this perform over time to sort of see if that's true within sort of an individual patient, which I think is a key question.
Yeah, well, so we didn't look into that. I think that's it's a very good question. This sort of, let's say, a prospective study will be very, very interesting. For practical reasons, we took a different approach of just taking a lot of samples that we had in storage and then looking at the clinical data in order to determine whether or not these patients are progressive IPF or stable IPF, and then analyze the gene expression data. It's very important to mention that the timing of the blood draw was on initial diagnosis when this patient, before this patient received any treatment. So this maybe leads to, I don't know, maybe an hypothesis that needs to be examined, that maybe this is of predictive value.
Great, thank you. I want to go back to Dr. Gooby. So you mentioned sort of following the exposures and sort of the decline in lung function. Were you able to pick up any of this is due to precipitation of acute exacerbations of any ILD, IPF, whatever the diagnosis was? I mean, does that predict at all what's going to happen?
So previous work by Dr. Kari Johansson and others has demonstrated the link between different types of pollutant exposures and the risk of acute exacerbations in cohorts of patients with IPF. It's a little bit trickier when we're looking at a broader cohort of patients with fibrotic ILD in order to clearly define what an acute exacerbation is in that population. So that does provide some limitation. We're currently in the process of collecting data on hospitalizations in our Simmons Center cohort, and we have some limited data from the PFF cohort on hospitalizations as well. And so instead of potentially looking at acute exacerbations, we plan on looking at respiratory related hospitalizations as a surrogate measure, because then we don't necessarily have to go into the weeds of trying to come up with a definition for acute exacerbations in fibrotic ILD that doesn't currently exist.
Yeah, well, good. So I wanted to put a plug in for the PFF registry that we've all heard about this week. This is now bearing the fruits of that labor of love from all of the people in the medical advisory committee for the PFF to put that together, because people like you are actually using that. So we should definitely thank them for that. We've got about a minute, so I have a quick question for Dr. Graney. So I guess the question would be related to where do you go next with your work in terms of caregiver burden? And you have 55 seconds or so.
Yeah, I'll keep it quick now. No, it's a great question. So I think it's really interesting. There's lots of different ways I see this research continuing forward. I think part of it depends on what we find with this study. Importantly, caregiver burden, as I mentioned, really has not been defined in this population. So part of it will be, do we need different questionnaires to evaluate burden for this population with pulmonary fibrosis or chronic respiratory failure? And then another part of this is going to be, how do we design interventions that target both the patient and the caregiver simultaneously that improve outcomes for both? So resiliency may be one way to do that, but I think it depends on what we see in this first step.
All right, listen, thank you all. Have a great weekend. Remember, there are sessions tomorrow to log in to see as well, and we hope to see you again at the next summit in Orlando. Take care, everybody. Bye-bye.
Thank you.
Thank you.
Bye-bye.